In my talk today, I'll quickly cover the definitions. I will talk a little bit about study designs that would be better for proof of concept evaluations, and then I'll talk about 3 different examples of how proof of concept evaluation studies could be done clinically, again, in the oncology therapeutic area. So with regard to definitions, a proof of concept is a clinical study which demonstrates early evidence of the potential for clinical efficacy based on clinical endpoints, surrogate endpoints, or biomarkers. And a proof of mechanism evaluation clinically is a study that would provide early evidence of pharmacological effect on a specific target or targets by the proposed mechanism of action. In a proof of concept study, we'd like to see evidence of efficacy based on the endpoints that I mentioned earlier. Decisions should be made based on predetermined criteria, and there should be a significant improvement in the probability of success of expensive late-stage clinical trials for a proof of concept study to be valuable. Biomarkers and clinical endpoints have been defined earlier. Biomarker is a characteristic that's measured and evaluated as an indicator of normal biologic processes, pathogenic processes, or pharmacologic responses to a therapeutic intervention. And a clinical endpoint, which is how drugs generally get approved, is how a patient feels, functions, or survives. So here is a tabular summary of the pros and cons of a single-arm proof of concept study, whether you call it a phase 1 study or a phase 2 study. In cancer, we basically could look at response rate in a single arm study. Results would be unequivocal with regard to the assessment of treatment effect. The study would be relatively quick and cheap. Assessment of response rate would be easy if the drug causes tumor shrinkage, and pharmacodynamic biomarker effects could be assessed. The cons of a single-arm study are that it is generally performed in a highly refractory population, so it may underestimate the benefit of treatment. It cannot distinguish between a subset drug, a drug that narrowly targets a population, and a drug with broad activity. In the absence of a concurrent control group, it's informative only for patients who have a shrinkage or response, so it may not be useful for assessment of stable disease, progression-free survival, or overall survival. And there would be issues with measured and unmeasured selection biases. It's not possible to assess the predictive value of a baseline biomarker using a single-arm study. It's always confounded because you can't determine if the value is prognostic or predictive. And it does not allow the assessment of biomarker in the setting of a combination treatment in a single-arm study. So going to the randomized control study, the pros are that it estimates benefit broadly as it measures effect regardless of the type of effect you are measuring, whether it's response rate, response duration, or progression, in everyone rather than a subset of patients with shrinkages. It provides protection, some protection from measured and unmeasured selection biases because of a concurrent randomized control group, allows evaluation of the experimental drug in a combination setting. Therefore, one could consider treating earlier populations because testing could be done on the background of standard of care. That is, you could combine your drug with the standard of care and have the standard of care in the placebo setting. Could be powered to recognize increases in the frequency of or severity of common adverse events, although that's not something that I have commonly seen done. And the predictive value of the stratification baseline biomarker can be assessed and, if appropriate, taken forward into phase 3 clinical trials. The cons of such a design are that it is not generally adequately powered to assure that the difference in outcome is always related to treatment effect and not a selection bias because it's a sample size question. These trials are longer, larger, and more expensive, and they're harder to sell to management as they're more time-consuming and expensive. So I'll now talk about an example of a proof of concept study using a randomized controlled design. It's an excellent way to evaluate predictive value of baseline biomarkers. However, the example that I have does not show that assessment, but we are doing some work internally to using this kind of design to assess the predictive value of baseline biomarkers. So this is an example of Avastin evaluated in a phase 2 setting. It was a three-arm randomized control trial. The standard of care for Avastin is 5-FU leucovorin. Avastin 5 milligram per kilogram or 10 milligram per kilogram was added to 5-FU leucovorin, and progression-free survival as well as response rate was assessed. Patients who were receiving 5-FU leucovorin alone when progressed received Avastin alone. And as I mentioned earlier, this is an excellent study designed to prospectively assess a potentially predictive baseline biomarker. However, no biomarker assessments were unfortunately performed in this study. Here is some data from that phase 2 trial, courtesy of Susan Hellman from Genentech. In this trial, the control group had a median progression-free survival of 5.4 months, whereas if you look at the total of the 5 and the 10 milligram groups, the median survival was 7.1 months. Median survival was also estimated for the control group, but was not estimable for the 5 and the 10 milligram group. And I have a note here at the bottom. For example, if one wanted to evaluate the predictive value of a baseline biomarker, such as tumor or plasma VEGF level, it could have been done well in such a study prospectively, as long as it was sized adequately. Here is some very interesting results. So as most of you know, in cancer clinical trials, benefit is assessed using hazard ratios. And so here is the hazard ratio in the phase 2 trial, which I just showed you, a hazard ratio of 0.54 for the two arms combined. And in phase 3, the hazard ratio was identical in this case to that of phase 2 clinical trial. So again, from a proof of concept perspective, this type of a design predicted the surrogate endpoint, the surrogate of survival, very well. It also predicted survival reasonably well, although it was not on the money. So here is my first example in terms of the a proof of concept study for evaluating a compound which targets the c-Met pathway. Here's a pictorial, a cartoon which depicts the c-Met signal transduction pathway, and as we understand it, there are amplifications in the c-Met receptor as well as mutations in the tyrosine kinase domain of the c-Met receptor, which signals downstream through the MAP kinase pathway and the PI3K AKT pathway. A number of drugs are under development for this pathway, and I'm going to talk about one of the drugs that's under development by GlaxoSmithKline, which has been published. In this study, patients with advanced papillary renal cell carcinoma were evaluated, and these patients, or some of these patients, have either a somatic or a hereditary mutation in the c-Met receptor, so the study was divided into two parts. In stratum A, patients with c-Met mutation, amplification, or polysemy of chromosome 7 were included, and in stratum B, patients without any evidence of c-Met activation were included. A daily oral 240 milligram dose of the compound was administered in a 5-day on, 9-day off schedule in this proof of concept trial, and response, safety, and biomarkers were assessed. Plasma biomarkers were also assessed in addition to the baseline assessment of the c-Met mutational status. So here is some data from this clinical trial in patients with stratum A. So I've drawn two circles here. Patients who have somatic or germline mutations in c-Met seem to have the greatest % change in tumor in this clinical trial. So the patients who have this mutation have a shrinkage ranging from 18 to 35%, whereas those who don't seem to have a lesser degree of shrinkage. The PFS data are not as clear, although the PFS data were not completely reported in the study when this was published last year. Here is some additional data in terms of response rate. So there were 3 responders in stratum A, in the stratum where patients had c-Met activation by one of the mechanisms, and 1 out of 16 in patients in stratum B. So in stratum A, clearly the response rate was much higher, about 33% compared to about 6 or 7% in stratum B. Again, perhaps confirming the preclinical hypothesis that this compound works by inhibiting the c-Met pathway.